Okay, thanks. Yeah, well, I'll start slowly and I hope a few people actually make it a bit later. Yeah. Okay, so this is what we're going to do. After a little introduction, I'm going to talk about active nematics, and in particular, active turbulence. And this is going to be the details of why you get active turbulence, the sort of systems that you get it in, and the sort of equations, the sort of theory you need to talk about active turbulence. And in particular, we'll get as far as active topological defects. And the blue bits will depend on timing, and I'm quite happy to miss them out. And it'd be great to have questions on the way through. And then I'm going to, in the last half an hour or so, move on to mechanobiology and ask the question, how do cells move collectively? And I think the answer is that we don't know. There's an awful lot we don't understand about that, even though it's a really simple question. You just have a layer of cells. How do they decide to move around? So I'll tell you about some of the phenomenology and then some very recent work we've been doing. But really, this is to ask the biologists in the audience help, what's actually going on here. Okay, so you all know about active matter. So we're all on the same page. What I mean by active matter is systems which take energy from the surroundings, and they do it on a single particle level. So each individual particle is taking energy from its surroundings and using it to do work. So this is biology. This is what biological particles do. They're always transducing energy, ATP or food, and using it to move. It's different from a driven system like something which you've been talking a lot about, shearing things. If you're shearing something, everything is subject to the same sort of force. This is a system which is pushed out of equilibrium, doesn't count as an active system. Let me start with some gentle movies to introduce the idea of active turbulence and the sort of systems where you see active turbulence. So this is the latest about how motor proteins move along microtubules. So this is a picture taken from, it's a computer rendering, but it's based on proper science. This is a motor protein walking along a microtubule carrying proteins in a vacuole. And this sort of system forms the basis of the most the best known and most useful system for looking at active turbulence, which is a mixture of microtubules and these motor proteins. The difference is that the motor proteins, which are kinesin clusters, have two heads, so they can bridge between the microtubules. Now, these things walk along the microtubules, and if they're all walking in the same direction, nothing's going to happen. But if one of them on this side moves that way, and one of them on this side moves that way, they're pushing the microtubules relative to each other. So the microtubules get stirred up, and if they're dense enough, what happens is that they tend to polarity sort, so all those moving down sort of stick together, all those moving up stick together. You get bundles, the bundles move, they buckle, they fall apart, and the process repeats. And you end up with a situation which looks pretty much like this, where those sort of white stringy things you're seeing are these bundles of microtubules, and they're being pushed around by the motor proteins. And the state you get looks like turbulence, in inverted commas, not inertial turbulence, but a chaotic state. And you do get structures. This thing here is a structure which is, say, repeated over there. So maybe we have to understand what's going on there as well. So another system is bacteria. This really, so I can show you the nice movie, is the motor protein, which works here at the tail of the bacterium. And this again is a computer animation based on the best science. And really, this is a slight non sequitur, but I mean, each of these different colored things is a different protein. Pretty amazing how nature can take all these different proteins and put them together to make a motor, and it's a motor which it can self-repair and make itself. I mean, we can't do anything like that using nanotechnology. So it's a pretty amazing result of nature based on active self-assembly. Anyway, when you take these things, these bacteria, and you put lots of them together, again, you end up with a turbulent-like state. This is strange because these are tiny. You're at low Reynolds number. Low Reynolds number flows are not meant to be turbulent. They're meant to be not chaotic at all. And yet you're getting this chaotic-like state. My third example is cells. These are epithelial cells. We heard about them from Rasko. And if you put loads of these together on a Petri dish and just look at them, as many of you know, again, these things can move like that. Again, you're getting turbulent-like chaotic behavior. You're getting something which looks like active turbulence. So what I want to do is explain this active turbulence and then ask, is it the same in all these different systems? And particularly at the end, we'll sort of look at these cells and say, do the bog standard theories of active turbulence work for cell layers? How might we need to extend them? So it's very much a progression, this talk, from stuff which is very well understood and is sort of the things you lecture on in summer schools down to questions where we really don't know the answer. So why do we care about this active matter? Why do we care about the collective properties of active matter? Well, I would have ordered these in a different way when we started working on this. I think people originally would think of active matter as an exciting new sort of engine. Can we take things like the microtubule motor mixture, put it in a microchannel, and have it do work for us? And people are certainly working in that direction, but it's hard. How do we harness this turbulent-like state to do anything useful? I think we're still at the stage where it's an exciting experimental system, but it's not actually useful for anything. The hardline theorists are thrilled with all this because we've now got lots of experiments where they can test theories of non-equilibrium statistical physics. These active systems are out of thermodynamic equilibrium. They're meant to be, and they stay there out of equilibrium. And so we've got loads of exciting new properties. And certainly, I'm sure in this school, you've seen a lot of different sorts of collective behavior. But increasingly, people are using these ideas to think about biology. It's a new way of thinking about biological systems, which if you're a theoretical physicist are very awkward because they're so messy, but they have lots of great properties. Can we use the ideas of active matter to understand what's going on in biology? And I put that first because now I think that's the most interesting and exciting question. And the jury's still out how much and how useful it's going to be. Okay, so this is really just a summary of these three different systems. We want to ask, what's this active turbulence? And is it the same across the three different systems? Please ask questions. Yep.
Turbulence in what sense? Is there some sort of a scaling similar to inertial turbulence?
No, it's turbulence in the sense of a chaotic state that looks turbulent. So the man in the street version of turbulence. I've got told off many times for calling it turbulence because that's meant to be Kolmogorov scaling and all that. But it's a chaotic state. Yep.
But I mean, I was wondering about that point. So if it's not the Kolmogorov scaling, so what would you give as a definition of when do I call it turbulent in this active turbulence sense?
At the moment, just that it looks turbulent.
people have tried to find scaling laws, and there are some scaling laws, but I don't think it's very important to what's going on in these systems.
Yeah, I mean, it doesn't have to be scaling laws, I guess. You could say, or maybe, I don't know, some characteristics of flow lines, or...
We'll see more of that in a minute,
Yeah.
but there is no theory of active turbulence. I think we understand what's going on, but it's more sort of looking at the pictures and explaining what's going on than actually a theory. So that.
Okay, so let's start with a nematic. I think we all know what a nematic is. A nematic is something which has this special nematic symmetry. It has a special direction, but up and down are the same. Things tend to line up with each other. And this is going to be the order parameter that I use to describe this nematic symmetry. It's a tensor order parameter. It tells you the direction of the order. Its largest eigenvector tells me the direction of the order, and its largest eigenvalue tells me how ordered these things are. And that's really a different lecture, so if you're not used to it, just remember Q is the order parameter. If you have a nematic, you can have a localized distortion like that, and this is an elastic liquid, a viscoelastic liquid. So a distortion like that's fine. It will just relax back very easily due to elastic forces because the minimum energy corresponds to everything pointing in the same direction. So these are easy distortions which are not a problem. However, you can also have topological defects in these systems. Distortions in the order which are much more important, if you like, because you actually can't unwind a distortion like these unless you change things all the way out to infinity. So this will change the pattern of the ordering all the way out to infinity. Defects like that actually can't happen in a perfect infinite system on their own because they would have infinite energy. So what tends to happen is that these things form near boundaries, or they can form in pairs. And then gradually the pairs will move towards each other and annihilate to reduce the energy of the system. In 2 dimensions, which is where we're going to be mostly, the important topological defects is this red one, which looks like a comet, and that has a topological charge plus a half, and this blue one, which has this threefold symmetry, and that has a topological charge minus a half. And again, I'm assuming you either know that or you believe me. This is a sort of basic course on liquid crystals, which we don't have time to do at the moment. You don't want these in your laptop because what they do is muck up the viewing angle, okay? So you hope they're just going to annihilate with each other and give you perfect nematic order. Right, so I've got 2 slides which are pretty hairy maths, but don't worry about the details. I've put on the details in case we've got any experts who want to see them. The only bit you need to worry about at all, and even that not very much, is the bit in the boxes here. What I'm doing is writing down the continuum theory for liquid crystals. So it's like the Navier-Stokes equations, but for these more complicated liquid crystal systems. So first of all, I need an equation for the order parameter. The order parameter moves with the flow. That's the normal advective term. And then this thing here says that it's turned by the flow, all right? Because if I have a liquid crystal molecule and I have any shear flow, it's going to be turned like that. So you need something in the equations to take account of that. And then this thing here is a sort of phenomenological term which says that if I turn off the flow, the system will relax to equilibrium. And the equilibrium that it relaxes to is described by the Landau-de Gennes free energy, the free energy expanded in terms of the order parameter. So here's the expansion in terms of this order parameter. It's a tensor with two indices. And then this thing here says that it really doesn't like gradients in the order parameter. They're associated with an extra energy. Okay, so that's the equation for Q, the order parameter. And then we need an equation for the velocity. And the way I've got it written here, this looks exactly like the Navier-Stokes equation. This is the usual advective term, the inertial term on the left-hand side, which is often small in these systems because we're at low Reynolds number. And here I have the derivative of the stress tensor. And in the stress tensor, I have the normal viscous term, which depends on velocity gradients. And then I have a horrible term, which is called backflow, which is the effect of the liquid crystal molecules moving on the surrounding fluid. So if these things turn, they're going to set up flows, and you have to include that in the equations. Yep.
In the energy you were writing just before, you have only one of the possible fourth order term. Is it because the others are forbidden by symmetry, or is it just?
I think you can map them all onto here because I'm in, hang on, this is 3D, I put it really like that. Because by the time you've got Qi, J2, Jk, Qkl, I think the Q squareds will work.
Exactly.
Yeah,
The square of a trace, and yeah, okay.
Yeah, I think it's probably because these things are traceless.
Yeah.
But, yes, and actually it really doesn't matter because this is doing the physics for you. This looks to stabilize.
Yeah, that's probably the answer. Okay.
So the really important thing is we can do it. We can write down the equations for liquid crystal hydrodynamics. This is well known. They're horrible, but if you need to use them, there are lots of codes which do it for you. But what I want to stress is how are these changed if you go from this passive system, no activity, to this active pneumatic. Why am I calling it a pneumatic, okay? Why am I starting with the equations of liquid crystals to see how active pneumatics are? How do I extend these equations to look at an active system? So there they are again. This is
The sort of basic idea, and I want to fill in this word in the title, active. How do I do it? Let's think about that. Let's look at move. Okay, let's look at this active turbulence again and say what's different about it than an ordinary passive pneumatic. Well, what's different is that now the pneumatogens are swimmers, they're moving, they're self-propelled, and so they're setting up flow fields in the surrounding fluid. They're setting up flow fields, and that's the extra thing we have to put in the equations. So what do these flow fields look like? Well, the flow fields look like this. This is one of these swimmers, and what I'm plotting around this swimmer is the far flow field. Okay, so if you measure the far flow field, and this is a brilliant experiment, a very famous one from Ray Goldstein's group, measure the flow field around loads of swimmers, organize your data so that the swimmers are all going in the same direction, and average, you end up with this picture, real experimental data. And what you'll see is there's some very obvious symmetry of the far flow field. And in particular, this symmetry is pneumatic, right? I've got a preferred axis, but forwards and backwards are the same as each other. The flow field around each individual swimmer has pneumatic symmetry. The maths version of that is that the reason it happens is that these swimmers are self-propelled. There's no force acting on them. And so if you do a multipole expansion, the leading term comes out from the force acting on the swimmer, which is 0, you lose that term, and you pick up the next to leading term, which is a dipolar term, and this dipolar term gives you this sort of flow field. So we need to put in the effect of that flow field in the equations of motion, and I'm going to, it would take me a lecture to do the derivation, which is due to Sriram Ramaswamy, and even then it's a slightly hand-waving, because in the middle he says, and now let's coarse grain, which basically gets you from the microscopics to the answer, but it's a very clever derivation, and the outcome is that you take those equations, and the extra thing you have to put in to take account of how these swimmers are moving is an extra term, which is zeta times q, which comes in to this active stress. That's all. That makes it an active pneumatic rather than a passive pneumatic. This very simple term, which comes in the stress. Yep.
Rototubules?
Yes, because actually the way I've got it drawn, you'll see that they're little arrows which were pointing. One was being pushed that way, one was being pushed that way. So the basic force structure is a dipole. It works exactly the same way. Cells, it's much less obvious. That's a big argument to say. Right?
Okay, so in terms of symmetry, it makes sense, right? I've said that this thing has pneumatic symmetry, which is why we've got a Q there. But I am, to some extent, although I'm motivating the answer, I'm not doing the maths, which leads you to the answer. But that maths is done to the extent that it can be done. And this is right, and it fits with experiment, and everyone believes it, okay? This zeta is just the strength. It's just a parameter, which is the strength of the activity, how many swimmers per unit volume and whether they're well fed or not. And then this thing Q is the order parameter. And the important thing to notice is that that comes in here and it comes under a derivative. So any changes in the order parameter give you stresses, give you flows. Any time the order parameter changes in an active system, either by changing its direction or by changing its magnitude, then you end up with stresses in the Stokes equation, and you end up with flows. Okay, so this I can use to explain almost anything that happens in these active systems. Let me just introduce a couple of words which I will probably use later, although I'm going to debunk them later. This zeta thing, if it's positive, you have an extensile system. If it's negative, you have a contractile system. And, sorry, it doesn't like it this far away. And an extensile system is one where the flows go in the direction of these arrows. In swimmer language, it's a pusher. Yeah, I think so. If I change the arrows over so the flows are going outwards from the sides and inwards from the ends, that's contractile. And in swimmer language, that's a puller. I've got those the wrong way, right way around. Okay, so symmetry of the flows ends up making a bit of a difference to what's going on. Okay, so let me just stress that thing in red. Right, that's really, if you want to understand what's going on in these systems, that's a really good place to start. So let's see what it does for you, this extra zeta q term, this extra stress term. I had this picture of a passive pneumatic, and I said that a little distortion like that would just relax back to equilibrium, no messing. However, it's a change in the order parameter, yeah? And so in an active system, it's going to lead to stresses and it's going to lead to flows. And if you work out the direction of the flows, it looks like that. And so this tiny little distortion is going to grow, and it's going to kill the nematic order. Nematic order is unstable in an active system. In maths language, if you do a linear stability analysis, you end up finding that the system is unstable to any deviations in the nematic order.
So is this more likely to be a bend instability, or?
It doesn't matter, because if it's an extensile system, it's unstable to bend. If it's a contractile system, it's unstable to slay. So you're always going to get some sort of instability, and it's always going to screw you.
But the type of instability depends on whether it is a pusher or a puller.
The type of instability that is unstable depends whether it's a pusher or a puller. I've drawn here a bend fluctuation, but there are always going to be fluctuations of all sorts in these liquid crystals.
Systems also, or only in infinite?
It's true in systems after a certain size, and I'll say something about confined systems later. In confined systems, the instability is cut off, and you can get stable states. Yeah. Okay. So this is the same story with a slightly more detailed picture. So if you're happy with that slide, the slide we just have to stick with it. This one here is the same thing again, and it shows you that if I have nematogens here, these are extensile nematogens, the black arrows are the directions they're pushing the flows outwards, and they're in a nice regular array, okay? So the flows are going to come out like that and then inwards like this. So these are the flows, and they'll all balance. And in this nice regular system, everything will be beautifully balanced. Then what happens is a tiny fluctuation. A tiny fluctuation means that I have a small bend fluctuation in the nematic system. And let me draw the flows. And what you can see is that these arrows are slightly more dense than those arrows, which is a pictorial way of saying there's a net flow in this direction there, and there's a net flow in this direction down here. And what's going to happen then is that this bend instability is just going to grow and grow. And you're going to lose your nematic order, but then you have to say, right, what do I get instead? Okay. Linear stability analysis is not going to tell you what you get instead. So what you have to do is to actually solve the equations. Before we do that, let's just have a look at an experiment where you can see this instability. So this is the microtubule motor system. These are experiments from Francesca Segue's group. And the problem about seeing the instability in experiments is getting the microtubules lined up to start with, so you can see them get unaligned. And they had the clever idea of sticking a straw into their system, right? And then they have a graduate student sucking up through the straw. I'm not sure if it's quite like this, but this is the general idea. So the graduate student sucks up through the straw, and so everything's going to align radially. The microtubules are going to be pulled radially because of the fluid flow towards the straw in the middle. And then you stop sucking before you get a mouthful of the stuff, and these things are going to do something, and we'll see that they undergo this bend instability. I'll play it twice because it happens rather quickly. Nicer here because it's cooler. There we go. Okay, so this is getting everything lined up. You can't see them when they're lined up. And there's the bend instability starting in the microtubules. Let me play it again because it's really hard to see that until you're ready for it. So there they are lined up, and here's the bend instability starting. You can actually see a secondary instability perpendicular to the first one. And then it's starting to become really turbulent. Yeah, Andres?
seems that there is a preferred length scale where this instability occurs. Is this clear?
Yes. There's a preferred length scale.
This is this active length scale here, and at least in terms of simulation, model parameters, it's the square root of the elasticity divided by the activity. Right, so we've got this far. Let's think about the active turbulence. And to look at that, we actually need to do the simulations. We need to take these equations and solve them on a computer. And if you do that, we're up to here, basically. Okay, let's look again at the experiments. You've plot the flow field in simulations. It looks like this, where the yellow bits are sort of streams of high velocity. So you tend to get these streams of high velocity. And these states in particular have high vorticity. Active turbulence is a very vortical state. This is the vorticity field. And the red bits are vorticity going that way, and the blue bits are vorticity going that way. And the length scale of these vortices is about 5 to 10 swimmers wide. So this is a very typical picture in all these systems which show active turbulence. Let me just take this picture and sort of zoom in so we can see a little bit better what's happening to the individual nematogens. I'll do questions a sec, okay? All right, so this is the vorticity picture with the flow lines on it. Let's zoom in. And what we're seeing here is also those little black bars are individual nematogens. Let's zoom in a bit more, because what you can see is a very obvious bend instability here in the nematogens. They're bent around, so you've got loads of stresses here where you have large gradients, and so you get flows, and it's these flows which are those streams of particularly fast motion. Yeah.
Questions? Questions, anyone? Okay.
So what about the topological defects? I've been arguing that this is a nematic. Okay, so if it's a nematic, we ought to have topological defects. So let's actually do a simulation and put a red dot where there's one of these plus a half defects and a blue dot wherever there is a minus a half defect. And these things are going to move around, pushed by the flow. And what you expect in a passive nematic is just that every time a plus and minus meet each other, they're going to annihilate. So let's see if you can see that happening. Those two did, but wasn't quick enough. Those annihilate. That's good. But then look up here. You get pairs popping out. And by the time this is finished, I'll play it again, the number of defects is pretty much the same as it was at the beginning. You're getting pairs annihilating with each other and also plus and minus pairs being created. So you're ending up with a gas of topological defects, which are continually being created and being destroyed. So does that make sense? Well, it sort of does, right? Because this system is active. So you're pumping in energy. And this is the energy which gives you the ability to create these defects, to create distortions in the directive field, which end up looking like a pair of plus and minus a half defects. If nothing else happened, they would then just annihilate again straight away. But the plus a half defects are motile, they're self-motile. this thing will move on its own. Anyone like to tell me why it will move on its own? Okay, well, you can get that just from this idea that changes in the order parameter give you stresses, give you forces. Okay, so the order parameter is changing, and so there are going to be forces on this system, and it's going to move. Yeah.
Looks like it has a specific orientation because it's looked like the tip of something. So then we would like to know if it moves. It should move in a way which corresponds to this orientation. But the other one does not have a specific orientation. And is it more immobile? I mean, less mobile than is the blue?
Yes.
Less mobile than the red?
Yes, yes. I mean, in a way, that was the next sentence, right? You're quite right. This has a direction to it. It's a polar particle. It has this special axis. And these things move towards their head in extensile systems, and they move towards their tail in contractile systems. This one here sets up flows because they're gradients in the order parameter. But these flows have a threefold symmetry, and you get balance between the flows. So you see flows, but almost no net motion for this sort of flow. So they're actually moving in two ways. They're being moved around by the background active flow, and they both do that. Then these plusor halves are also self-propelled. And by looking at the direction of them, that's a way we've used to decide whether the system is extensile or contractile. Okay. Yep. Because they're formed in pairs, and it doesn't take an infinite energy to get a pair of defects, because the distortion is fairly local and it doesn't go all the way out to infinity. So it's because they're in pairs. You certainly, if we look again and you see them popping out, move. Sorry, I'll go back to the beginning and get that under control. You'll see that they're always popping out in pairs, plus or minus pairs. I'm used to looking at this, and there's two K in pairs. I think up here you'll see them pop out, and then the red one zooms off. Yep.
You only have spontaneous creation of plus half and minus half defects. Can you not have like plus 1 or minus 1 defects?
Yes, really, because the energy barrier is so much smaller. So they will appear before you can get enough energy to give you plus and minus ones. There are plus or minus ones even in various systems in nature, like hydro particularly, poster on that, but not here. Yeah.
If you run the simulation with a spherical topology, you expect to get like a minimum charge, more red than blue or something like that?
That's right. Vasco's the expert on that. People have done that sort of physics, that's right, and you get a +2 net charge on a sphere because of the topology. And sometimes it's 2 + 1s, and sometimes it decides to be 4 + a half. Okay, so what else were we going to say? And this is the experimental version of the formation of these topological defects. This is the microtubule motor system. Remember that the microtubules tend to buckle and bend. So this is microtubules which are buckling. And what you can see is that the buckling is essentially the formation of this +1/2 defect, which is this comet-like structure, and the -1/2 defect left behind has this threefold symmetry. The -1/2 doesn't move very much, and the +1/2 moves away from the -1/2. So you're seeing the same physics happening in the real system. Yeah, in the real system. Okay, so now let's see what happens. Partly so I can show you some nice movies. If you take this active system and you confine it. If you confine it, it cuts off the instability. And when you have a confinement which is of order a few microns, that means that these vortices, which characterize active turbulence, don't form. So what you can get instead is much more regular flows. For example, this is what happens if you put the microtubule motor system in a long, thin channel. You obviously can't just get flow along the channel because of what happens at the ends. What you actually get is a shear flow, and you get it going, shearing like this, and then sort of turning itself round at the ends and moving back. And here you can see a regular shear flow being set up. Then occasionally you get topological defects coming in. Sorry, they have a different color scheme in their movies. Greens and blues are plus or minus halves. You get these sort of clusters of topological defects. And the reason for that is that the shear flow is pulling the microtubules out so that they're stretched. So they're stretched and nice and pneumatic. But pneumatic's really bad if you're an active pneumatic. You get an instability, and what happens is that these things bend, and these bends look like pairs of topological defects. So if I play it again, you can see that it starts being a nice shear. Starts being a nice shear, and then microtubules are being stretched out along the walls because there's a slip boundary condition, and you get these bends which can be identified as defects. Yeah?
A quick question. If to get this stretching effect, do you need a particular channel to characteristic size of the tubule ratio?
No, but to get the regular flows, you need the channel to be small enough that it stops active turbulence. Okay. Yeah. And you would need slip walls on your channel to do that, right? This is the same thing
happening now in a circular confinement. Comes out a beautiful movie. Again, you get, now the regular flow is a circular regular flow. Everything is pushed around, but then occasionally you get an instability and a topological defect is formed. And we can now see exactly this in the simulations. Cute movie. And of course, macroscopic active matter does the same thing.
Are there any specific ratio between plus and minus defects which can be defined by confinement or by geometry of the system?
You essentially have to have equal numbers. You always have equal numbers of pluses and minuses. The only slight caveat to that is that you can have sort of virtual defects just beyond the channel boundaries reflected in the
Nematic line just inside,
which you have to count because it's equal plus or minus. It's this thing that a single plus defect can't exist because it hasn't.
I think I'm missing the microscopics of how this bending is happening. So if I imagine these microtubules to be a string, if you stretch them, I would imagine they are under tension. So it's difficult to bend them.
It's not individual ones. It's actually bundles of these things, right? And what you're killing is the pneumatic orders. So what happens is that the minute they bend a tiny bit due to a tiny fluctuation, they're setting up flows which are pushing them to bend more. And that's why they're bent like that. And that wins the shear flow, which is trying to elongate. I think you're right that if the shear flow was very strong, you wouldn't see it.
All right, so that's the conclusions to this bit of the talk, and I put a couple of reviews there if anybody's interested in learning more about this. I guess this is quite old now, but it has all the basics in. And this is nice because it's a collection of articles on all sorts of active matter, and they're only 4 pages long, each of them. So it's a really good lead-in to anything you want to learn about. So what am I going to talk about now? Okay, so now the next bit is sort of advanced topics, if you like. So I'm not going to go into any of them in detail, but just show you a couple of things. And the first one is to ask the question, are there any other sorts of instabilities in these active systems? Because I've talked about systems which start with pneumatic order, and then that pneumatic order is killed by the active instability. But if you think of something like microswimmers and you turn off the activity, they're not going to form a pneumatic state, right? So the limit I start with is really not right for these swimming systems. If you turn off, yeah, sorry.
Question, because I don't understand nothing. So, what is the source of energy of these swimmers?
The swimmers, it's ATP essentially. Right. It's a bit biology for me, and it depends on the system, but the biologist said they would tell you that it's mechanical.
So no temperature, no, just...
No, it's chemistry. And these things you can sort of forget thermal fluctuations. You're at a larger length scale where you don't expect thermal fluctuations to be important.
So when the food finish,
you have to keep feeding them, yeah.
Okay.
Yeah, it's not perpetual motion, unfortunately. You do have to keep feeding them, yeah. But you're right, that's what I want to worry about. You know, if I do turn that off, that source, are they going to line up as a beautiful passive pneumatic? And the answer is no, right? They're all going to fall into a horrible heap. So let's start off with a system which is a pneumatogen, a load of pneumatogens, but they're not ordered. They're isotropic. So it's like having a liquid crystal at infinite temperature. They're still long and thin, but they're randomly ordered. And say, can I get active turbulence in that sort of system? Okay, and the answer, I'm worried this for a while, but the answer is yes. And it's yes because of something which I've called instability 2. Right, so this is quite tricky, but there's just one slide, so don't worry too much if you get lost. So I start off with a system which is a load of pneumatogens, like this bit here, which are pointing in random directions. All right, so the order is 0. The net order is 0 in this system. And then I have a tiny fluctuation which orders them locally. Now, for the point of view of this graph, I've ordered them lots, so they're really beautifully ordered, but you only need a tiny bit of order so that S is non-zero in these regions. These are extensile pneumatics, and so what's going to happen is that this order is going to set up flows. The red arrows show the flows. The flows are pushing outwards along the long axis and inwards along the sides, and similarly here you get these red flows set up. So what's going to happen to the ones in the middle, where there wasn't a fluctuation, is that they're under a shear flow. Yeah, so these guys are going to be subjected to a shear flow. And so what's going to happen is because they're subjected to a shear flow, they're going to tend to turn around and point in this direction. So they're going to tend to line up with the ones which are already aligned due to the shear. And so what you're going to do is get this sort of bootstrap effect, that the minute you get a tiny bit of pneumatic order, it will tend to bootstrap more pneumatic order. And you can show that
using the equations as well, that works. So what's going to happen is that if you start off with a system which doesn't have nematic order and you turn on activity, the activity itself will give you nematic order, which will then be killed by instability 1 to give you active turbulence, and you're going to get this sort of cycle where the one instability is busy making it a nematic, and the normal active nematic instability is destroying the nematic order again. And that is probably what's happening in these bacterial systems, and it's certainly what happens if you run the simulations. Right, so that's instability 2. Now we've got instability 3, which is even, which we really don't understand properly at the moment. And that asks the question, which we'll return to in a minute, but you know, these cells, right, these are MDCK cells, these are squishy cells, and on average, those are not nematic. They're not long and thin particles, they're circular, squishy particles. So why on earth am I talking about active turbulence when I'm starting with circular particles? Why can they be nematic? And the answer is, again, you get another instability, which basically takes these squishy particles and elongates them. Let's have a picture here. Because they're creating flows. Some of these flows will be extensional flows. So a circular particle will tend to be extended, and then it will pull its neighbors. It's connected to its neighbors, so it will pull those, so those tend to be extended. And so that's fine, because then you've got a nematic, at least locally, and you can get this active turbulence. So it's okay starting with circular cells as long as they're extendable, as long as they can be pulled out to be a nematic. Right. And then the other thing is just to show you this for fun, I think. Right, so this is sort of out of the main development of what I'm going to say. But this is some experiments, or I should say who actually did the work here. Experiments, Harandu and Yilin Wu's group have done the experiments, and then Marana in my group has done the simulations. So this is an example of this instability, and it's instability 1 now. So this is what we're doing. This is a blob of bacteria on a substrate. So it's P. mirabilis. This is about 500 microns across. They don't move much in the center, but around the edge they do move, and it looks like they're doing active turbulence. And what happens is that, let's see if I've got the right movie here, yeah. This thing organizes itself so you get these regular protrusions around the interface, and then the protrusions burst, and you end up with these sort of streams of bacteria escaping from the drop. So now let's look at these escaping streams in a bit more detail. So this is the edge of the drop, and you can see the protrusions start to grow slowly in this movie and then burst. There they are bursting. So we're now looking at a smaller length scale, and you get these beautiful structures forming. Right, any questions? Was I clear about the system? Do you have questions? Okay. And I claim that this is this active instability working. And the reason I claim that is, first of all, let's just think about the big drop itself, and then we'll think about what's happening in these rather lovely channels which are formed as the drop expands. So if you measure, this is the protrusions at the edge of the drop, and if you measure the spacing of these protrusions, it's definitely peaked around a certain value. This is a real experiment, so peaks and width to them, but you tend to get this sort of spacing between the protrusions. They're regularly spaced. And if you do the simulations, what you can see is that you get this active instability forming at the interface. And so you get regularly spaced protrusions sticking out. The ordering within the protrusions looks like +1/2 defects, right? These things are like +1/2 defects. And so that means they're going to be forces pushing them outwards. And so you get the formation of the protrusions and you get forces pushing outwards from the drop. And that's what's forming these regularly spaced protrusions. And some of them burst. That's just physics of the pushing makes them burst, and the the bacteria can move out. And when the bacteria moves out, you end up with this sort of structure here. And again, what's happening with those structures is that the long, thin channels are undergoing this active instability. And so what's happening is that you're getting, again, these tiny protrusions on the edge, except it's a mess because there are lots of them together. And again, you can look at the scale of the distance between these protrusions and show that that fits with thinking of this as an active instability. So it's, I think, a rather cute example of active matter physics in action. And I'm happy to talk about that in more detail. I mean, I just told you, I can show you the evidence from what I've said. But I prefer now to move on to this mechanobiology thing, because I'd really like people's input on that. Yeah?
The distance between these
protrusions, yeah.
Okay. If you increase the radius of the drop, also the distance increase between the protrusion.
That shouldn't, well, as long as the drop's quite big, no. But if you make the drop smaller so the curvature matters, yes, the curvature makes a difference. We think that the reason the curvature is making a difference is that this is an active instability which is trying to bend stuff, but it's already bent in a curved drop. And that means that the protrusions tend to be slightly closer together. And that works in experiment and in simulations.
Thank you.
Yeah.
These active protrusions that you see, are they specific to these type of bacteria, or is it universal?
It's this sort of bacteria. I mean, they might happen somewhere else. Certainly the first thought was this is something to do with fractals, because that pattern looks fractal-like, and it isn't. It's been seen more than once in the system by 2 different groups, so that's okay, but it is very much bacteria dependent.
Any understanding why in these particular bacteria you get this particular phenomenon?
I think part of it is that, in fact, in the middle of the drop, because there's less oxygen, they're not moving much. So really, it's a ring of motile bacteria. And that means that if it was the whole drop, you'd get active turbulence everywhere, and you basically just get a mess. Because it's a ring, it's much more like active turbulence in a channel, so you can get these stabilized drops. So I think it's luck to some extent. I don't think this is sort of generic, this is how bacteria all this thing. Okay, so now let's move. Let's actually do real biology. Apologies to any real biologists in the audience.
Please ask questions because I'm learning biology. And it's hard. Okay? So the question we are trying to ask at the moment is, how do confluent cell layers move? So let's start by saying, how do individual cells move? And if you're a physicist, the way you would model how an individual cell moves, which works really well, is just that you have a polar driving. By polar, I mean just a force acting on this thing, all right? And then because it's a squishy particle and you don't want it to leave its tail behind, you'd also put on some sort of contractile force. This would be a typical contractile activity like you get in active systems, which will just pull in the tail. And people have done loads of models based on more complicated versions of this, and they work very well. That what a single cell does is a directed, persistent random walk. Okay, so you put noise on this and you end up with cells moving. If we want this sort of level of description now for layers of epithelial cells, both those taken out of the body and put on a substrate, and also those eventually which are moving inside the body. And the sort of cells I'm going to think about are these epithelial cells, which Rasko told you about. Okay, so just for those of you that are not biologists, why am I saying that cells move inside the body? All right, because they pretty obviously, our cells are not sort of wandering around in a sort of horrible extraterrestrial way, all right? But there are some very important times when cells do move. Wound healing is one of them. The cells have to move to heal the wound. Very important time is right at the beginning of life, when you start from a tiny little cell and the cells grow, but also you have to end up going from a little ball of cells to a human being, and therefore they're really very beautiful concerted movements of cells. They suddenly take off and move in various directions in a very complex but very well orchestrated way to form gastrulation, embryogenesis, and transform you from, say, a tadpole to a frog. Then there's cancer when cells suddenly decide to take off, often as single cells, but you can also have collective cell motion. And very slow cell motion, like in the skin or in the gut, where cells in the skin move always up through the skin to form new layers. So actually cells do move, even though they don't look like it to start with. If you take them out and you put them on a Petri dish, then they move like mad and they do this thing which looks like active turbulence, unless you put them on a different sort of substrate, in which case they don't move at all. And I think that's not well represented in the literature, just how much difference it makes, the different ways these cells, it really matters where you put them. Yes, sorry.
What we see are cells dividing?
The white spots are actually cells dying, so they're being pushed out of the layer. Yeah, yeah, and we're sort of ignoring that. Yeah. Okay, so let's start with cells which are not, mostly not in the body, and ask what are the different ways these cells can move. Well, one thing they can do is flocking. Okay, so flocking is they will take off and move in the same direction. This is the cells inside the egg chamber of Drosophila, which happily move around like this for reasons which nobody really understands. This is the same movie saying the same thing of the cells moving. So flocking definitely happens. Okay, and flocking means they all move in the same direction. And in vivo, you see flocking as well. A lot of the early gastrulation, embryogenesis things are groups of cells which suddenly take off and move somewhere else. Then, perhaps surprisingly, you get this active turbulence. I've shown you pictures of active turbulence. We'll see it again. Why do I think that's active turbulence? Well, if you look at the velocity fields, one of these is from our simulations and one of them is from a load of cells, MDCK cells. And these are topological defects in another sort of cell. And they move around and they annihilate and they're created in pairs. And the topological defects look pretty much like what's happening in the cells. So it certainly makes sense, at least, to think about these things being active turbulence.
And a really beautiful experiment from the Paris group is that they took cells and they put them in a channel. So they've got them confined. And so what you expect to see is this regular flow because they're confined. And what they saw indeed was a regular shear flow, which they don't have a nice movie of. But what they did, which was lovely, is match it to the theory. You can solve exactly what's happening in a channel. You can solve for the flow profile of an active system in a channel, and it matches these cells pretty much exactly, which was a lovely justification that these are acting as an active nematic. Yeah.
Know what is the individual director of a cell, which is maybe not trivial.
Why? I mean, what they match is the velocity field, yeah?
Okay.
Really?
So you're not solving for the magnetic order parameter.
I think they could. I can't remember whether they do, but you can do the individual director of a cell. You can segment these cells and you can look at the shape of each cell and apply, yeah, the shape. But what I'm going to say later is that the shape might not be the right thing to look at. But that's a different story, really. And then this thing here, okay, this is, it suddenly decides it's being confined. And so rather than active turbulence, it decides to go around and round, like we saw in the microtubule system. And this is some experiments from our original paper on this, a long time ago now, but I still think this is a cute experiment. This is by Van Ben-Soor and Ben Leduc's group did the experiments, and this is a plot of the number of topological defects as a function of time. And here they added blebistatin, which basically sends the cells to sleep. They don't want to move, stops them being active. And the number of defects goes down because it's acting as a passive system with defects annihilating. You wash out the blebistatin, and the number of defects goes up. And we then looked at the topological defects in this system. Here they are. Here's a plus a half defect, and it's moving towards its head. Moving towards its head. And hang on a minute, that means the system's extensile. Cells just get longer and longer. But that can't be right, because cells, we've said, are contractile. The machinery in a cell wants to take an elongated cell and return it to circular. So that worried us for ages. It still worries us, but I think we maybe have an answer to that one now. We spent ages trying to work out how cells are extensile, but I think we learned a lot, but I didn't think as much to do with cells. Now I'm going to study Roberto's beautiful experiments. I love these experiments. I still think these are the most beautiful experiments out. All right. Apart from anything else, the data actually looks different in the two cases. So what he did is he plated this sort of cell, which is, again, an epithelial cell on a substrate. And it does something which looks like active turbulence. And this blue line shows the velocity as a function of time, the average velocity. So these things are sort of dotting around, doing active turbulence happily on their own. Tell me if I'm getting this wrong, okay? Yeah. And then they added a chemical Rab5A, which probably mucks up the junctions between cells. No one, at least last time I asked him, was quite sure what it does. And what happens is these things just take off and they flock. You get much larger velocities in correlated directions. So add Rab5A here, the velocity goes up, you get flocking, and then gradually goes down to where it was before. And this is a movie of the same thing taken from their paper. This is active turbulence. That one again, it's going to work from here probably. And there you can see it is flocking. So probably the only thing they did was change the interactions between the cells. And another paper which I like, which is a
much more recent paper from the Princeton group, from Daniel Cohen's group in Princeton, is that they took this MDCK layer. So it's an MDCK layer, and it's plated on a substrate where half the substrate is covered with extracellular matrix, and the other half is covered by E-cadherin. And cells are meant to cling on to E-cadherin much more than extracellular matrix. And what happens is that the cells, cells on one side of the substrate, this one, do active turbulence. They move around, they're busy doing active turbulence, and then there's a really sharp boundary, and these guys don't move. So it really matters what sort of substrate you've got. Okay? And I've been going around saying MDCK cells do active turbulence, but then talking to Guillaume Cheraz in London, that's true if they're on the right sort of substrate, this sort of substrate. But if you suspend them, so they're no substrate at all, they don't do active turbulence. They just sort of sway gently around as if they're being pushed in the wind. And certainly when I was talking about this in Boulder, there was Margaret Gardell says, my cells, my MDCK cells don't do active turbulence. They just sort of sit there. And again, different substrate. So, you know, we really don't understand this, what's going on. Sometimes you get flocking. Sometimes you get active turbulence. And I should have said jamming. When the density gets higher, you get jamming. And, you know, that's a very basic question. How do we model these things? There are all sorts of questions. I've listed some of them there, right? How can we understand what's going on in these systems if we can't even write down a very simple model? If we don't even know if the important forces are the forces the cells are extending on the substrate, obviously that sort of matters, right? Because substrates make a difference. Or whether it's the force between the cells where they're pulling on each other. These things definitely have active junctions between them, and presumably the active turbulence is due to cells pulling on other cells. So let me spend the last 15 minutes. Anybody want to make any comments on that, especially biologists? That's the right sort of questions.
Consider that the active turbulence come from a local disruption of the equilibrium, right? If the cell is dividing or is just popping out because it's dying. And then you think that the rupture of order comes from there, and then you have this.
No, because you can kill the cell division and you can look on the timescale short compared to cell death, and you still see active turbulence. My best guess about what causes the active turbulence is either that the cells are actually pulling on each other, so they're sort of squishy cells, right, and they're pulling their neighbors. And that force must be nematic because the cells pull on each other in a reciprocal way. So that would be possible. Another possibility, which was pointed out by Cristina Marchetti and also people in Imperial, was that it's actually polar forces, forces pulling on the surface, and normally forces pulling on the surface would give you flocking. But if these forces fluctuate quickly, then on average they'll look nematic-like because they'll cancel each other out apart from a dipole. So either of those are possible, and I don't think we or anybody else has really looked at the differences, and that's something we want to do. Yes. It's not really, the right experiments have not been done. I mean, some have been done, so, what you're talking about is T1 transitions, which is when cells move relative to each other. In our continuum models, you can't see those because you're not on cell level. In the models Rasko is going to talk about, the vertex models, you can see them much more nicely, and you can see them in the experiments, but nobody's really looked at them in experiments because they're a bit boring for biologists. Yeah. Yeah.
active movement of cells, even if they are overgrown, which usually can stop their movement by contact inhibition, might be also caused by their wish to get more nutrients and oxygen. So, do you know if anybody already tried to kill, like to put the cells in a very like low nutrient media and study this?
Not that I know of. I mean, they're just not biological questions, right? Because biologists don't care about it. But this idea of contact inhibition of locomotion, I mean, I've tried very hard to look at the literature, and it's a folktale. It's an interesting folk. I mean, it's just, there's nothing proven about it. It just looks like when you put cells on the substrate, it looks like they're not doing this polar motion. They're just sort of sitting there wriggling about. So what people say is that because of the contacts, they're not moving in a particular direction. But no one's quite sure what it is or why it is or what it means. Yeah.
It's possible also to observe what's happened during the transition between the constrained state of the particle and when they move. Yes, sorry.
So you're saying, is it possible?
If you show maybe just a few ratio of the total cells has this capacity of connected to each other.
Oh, so you're saying as it becomes confluent, what does it do? Is that right? You mean when you get blobs?
Oh, yes, just a few of cells.
It looks like the cells on the edge behave in a different way from the cells in the middle. So having a boundary just makes life more complicated.
Okay.
Okay, so I always feel that when I say this, I ought to the audience say, well, everybody knows that, but I don't. Okay. So what I really want to concentrate on is this idea, are cells extensile or contractile?
Because the way the topological defects move makes them look extensile, whereas everyone says cells are contractile, they want to return to round. That's what their biological machinery does. So this is work by these people, in particular, Jacob led the experiments and Marana did all the hard work on the theory. The punchline is that we shouldn't be calling cells extensile or contractile because they're squishy cells. They can be either, and they can change from one to the other. And so it's just not the right way of thinking about them. And the fact that these cells are deformable, so individual cells can deform, I mean, they could be stretched that way, they can become circular, and then they can be pulled that way, is different from nematogens, which have a fixed shape. So let's look for the evidence for that, and let me talk about first what I'm doing. This is the experiments they did. This is MDCK, same pictures we saw before. They measured the shape axis of each cell, i.e. the long axis, what you normally do for a nematic. And then they measured the direction of principal contractile stress, i.e. the direction the cell is pulling inwards most strongly. So we're assuming, it's an assumption, that this is a nematic-like force and that the cell is pulling inwards in one direction and pulling outwards in the other. And the axis is the axis along which it's pulling inwards. So normally, if you have a shape like that and a stress like this, the forces are like this, that's what you would have called, I don't remember which one, puller. It's a contractile particle, yeah? And that's all there is to it in terms of swimmers. Okay, so the same with a cell. The shape's like this, and the stress is like that. The thing will get pushed back to circular, and you'd call that a contractile particle. But because it's squishy, you can keep pushing, and what's going to happen is going to become circular and then get pushed out in the other direction. And then you've got something where the shape axis is like this. The shape axis has changed, but the stress axis hasn't changed. And you've gone from a cell which you would have called contractile to one which you would have called extensile. So let's forget about these terms, contractile and extensile, which are a pain in the neck. And let's look at the shape axis and this stress axis, and just define an angle between them, which I'll call the misalignment angle. That angle would be 0 if it was a proper contractile system. It would be 90 degrees if it was a proper extensile system, but can it be something in between? So this is what the experimentalists measured. And what they found, okay, is that this angle can be anything, okay? So what I'm plotting here is a region of this MDCK cell layer. In the red regions, the cells have a misalignment angle between 0 and 45, okay? And in the blue regions, they have a misalignment angle, which is big, between 45 and 90. Typically, this length is 4 or 5 cells across. So you get patches of large misalignment angle and patches of small misalignment angle.
And those patches evolve with time according to, this is a time sequence. And now this is a movie showing how these patches evolve with time. So in the blue patches, there's a real angle between shape and stress. Okay?
So shape is shape.
Yeah.
But stress, how is it measured?
It's traction force microscopy.
On the substrate.
On the substrate, all right. So you might worry. And then we assume that the stresses are dipolar. So we assume it's behaving like an active nematic, which is an assumption. And I would worry like mad, except our simulations where I know what's happening, get exactly the same thing.
Yeah.
Experiment itself, is the monolayer confined, or this is just like you see the whole monolayer, that's just the diameter of the...
It's not confined and it's not dividing, so it's just sitting there.
So like the circle would just be the well.
It's a bit in the middle, which we just happen to look at, yes.
I was confused by the fact that if there is nothing around, then they will go somewhere, right?
So
that's why.
They tend to stick together. These things really like to stick together. So if you do have a blob, they will stay being a blob. They won't suddenly fly off.
Yeah. Okay, so for visual purposes, we've done 0 to 45 and 45 to 90, but this is properly the distribution of this angle. You get this distribution looking like this. On the whole, the misalignment angle is smallish. This seems to be normal, but then you can get big ones as well. And this is just lots of different, I think we've got 11 different bits of 11 different monolayers, just to show we get the same answer lots of times. Right. Okay, so, yeah.
Found in the region of boundary between these two regions?
Good point. I mean, that's why I first started looking at this, to see why the defects form in the boundaries. We have simulations which show the same thing, and we're still working on an explanation I can stand here and give properly, so we don't really understand why. But yes, defects do tend to form in the boundaries. Only plus or halves form in the boundaries. I'm not quite sure why, but hopefully we're close-ish to. We can at least reproduce it, even if we don't understand it properly yet. I think it's something really quite subtle to do with how things align in the flows. Okay, so how would we write down, how would we take this bog standard theory and change it to match, to reproduce the simulations? Well, what we need is, the main thing is we need 2 order parameters, yeah? One which would be an order parameter for the shape nematic, which is allowed now to be different from the order parameter for the stress. So we've got 2 formulae, one for the stress and one for the shape. And may I say now to all biologists that I understand shape, everyone understands shape,
but I'm not sure when we measure stress, you know, we know mathematically what it is, but I don't understand physically, you know, am I doing the stress fibers in the cell? Why can they suddenly decide to move? Don't get that yet. Okay, so that's how you do it. And then we drive this with an active force, but we don't drive the shape. So just the stress is giving you an active force. And then we also, somehow we have to make the stress and shape respond in a different way to the flow. There are lots of ways of doing that. The way I did it in these simulations is to make the elastic constants a bit different for the stress and the shape. And then also these things seem to align with each other again. So if you get them misaligned, gradually they will align. That comes out experimentally. So we also put a term in the free energy which makes them move back and align with each other. And if you do that, these are the experiments, these are the simulations. It really works beautifully. You know, so I was worried about the experiments because they're really hard experiments, and the interpretation of traction force microscopy is a bit iffy, in my view. But it works in exactly the same way. You get these blobs, these misaligned blobs. So we've still got lots of questions, right? The model is not ideal. The reason the model is not ideal is that this particular model, if you want the stress and shape to change relative to each other, they have to turn. This model is for fixed length nematogens. It's got two order parameters, so shape and stress can be different, but they have to turn. What we're trying to do now with Sumesh Tampi, and Sumesh has done a lot of this now, we're almost there, is to allow the direction to change because the cells can go circular and then expand in another direction. We end up getting essentially the same results, okay? But it's a bit easier to understand how the parameters match onto the experiments. But the really big question here, so let me finish with the really big question to me, is what is actually happening in these cells, okay? Maybe the flow is changing the shape. The flow is pulling the shape. That makes sense. You get extensional flows which push it like that and then pull it like that. But what's happening to these stresses? You know, what are they, the stress fibers in a cell, or something to do with how the cells are pulling on each other? I really don't understand what I mean when I talk about stress in these systems. I understand in the simulations what I mean, but I don't understand how that matches to the biology. And that's what we're trying to think about at the moment. And I also should say that Benoit Ledoux's group has got almost identical results, which makes me happier because it's been done twice now. That's probably a good time to stop. That's what I've been saying. Thank you very much for listening.
One more question. I think you consider the monolayer of cells having the same rate of division and death?
They're suppressed, so we're doing it on a time scale where division is not very important. They've now put division into these experiments.
And dying cells too?
I doubt it, actually. I think it probably is making them. And you see the same thing. You see it become a little bit more, the contractile-ish regions, the misalignment gets smaller. That doesn't really fit with this theory because cell division acts like extensile stress, so it makes it more extensile. But it does fit with this new theory we've got with cells being squishy and how they are pushed, because dividing cells push things in certain ways. And the reason I'm being a bit iffy about it is because this is all sort of super new, like sort of stuff.
I was just wondering if this force of the whole monolayer of the whole group of cells could have some additional parameters, such as the little cavity which is generated for some seconds after a cell is dead. it dies, it grows up, and the little cavity is generated. And this attracts every time some new cell from around, from neighbor, going into it, filling. And this might change the direction of originally like something which goes automatically in.
I mean, you're right that we have done, not relative to this, but just looked at dividing cells. And a lot of people have done this. Actually, is looking at flow field around a dividing cell is extensile. Dividing cells make things more extensile. And dying cells, when they leave the monolayer, I would expect to make it more contractile, although people haven't done those experiments. But here, there is no cell division to speak of.
I have a question instead. So in the experiments that you showed, our experiment, we also monitored something a little different from what you just did. So we labeled some of the lamellipodia, the critical lamellipodia, which are the structures that the cells use to exert those stresses that you mentioned. And we basically looked at the correlation, at the angle between the direction of the lamellipodia, which is basically the same of your stress, and the velocity of a group of cells around that cell. And we found that there was an isotropic distribution in the movie on the left, and there was a distribution with a peak at the frontal direction for the flocking one. We did it because we wanted to check whether there was flocking or not. This is not what you have done, but if you have to speculate what will be the result of this analysis on yours.
I mean, I think that makes a lot of sense, that we assume that there's a polar force, which is what you're saying, a directional force for flocking. And I still don't understand. I mean, I thought it was just cells pulling on each other through junctions, which would give active turbulence. But what would work just as well is actually an isotropic distribution of forces, which are chained with the substrate, which are changing quite quickly on the timescales the cells move. So I had sort of said contact inhibition of locomotion turns off lamellipodia, but I am now thinking it randomizes lamellipodia, which is what you're saying.
I mean, we see them. We see them also in the controls.
But how big are they? Are they smaller or just?
No, they just move in random directions.
Randomly. Okay.
They are extraflected here, and then here, and then here. When they want to flock, they are extraflected exactly in the direction where they want to go.
Yeah, I mean, I think there's a lot we don't understand. Also, these experiments by Guillaume Charraz, which says that if they're linked with each other with no substrate, you don't get active turbulence, is making me think that actually we should have thought of it much more as a fluctuating polar force. And we want to do the simulations to find out the difference. Yeah, yeah. It's actually you showing me this cryptic lamellipodia, which made me worry about that as well. Yeah.
Other questions? No? So let's